Welcome back to another Amino Labs Bioinformatics session, where today we're going to use some bioinformatics software called FIJI, also called ImageJ, to do some image analysis of Agarose gel results. Now, the reason we're doing this today is because we want to try to estimate how much DNA is in a particular band on a gel that we've run. And maybe you've done a PCR reaction and you want to know how much DNA is actually in your PCR reaction. And so that's our goal today. If you happen to be in a big expensive lab, you might have access to some high-end equipment. In particular, a spectrophotometer is used to generally calculate the amount or concentration of DNA in a sample. But because you don't have access to that, we're going to try out this method today. All right, so you're going to need a couple of things here. One is this spreadsheet, which you'll find a link to on Amino Labs' website, amino.bio. You're going to need to download some software called FIJI. And again, this is an open source science suite. It's been in development for years and years, and it is used by so many research labs around the world. So this is a great opportunity today for you to practice using software that real labs are using very, very often. One thing you'll need to quickly get familiar with, you might already be, is the DNA ladder. And you might remember this because if you loaded your gel in your sample, you will have loaded some ladder. And we're going to talk about that a little more today. And you're going to need your results from running a gel, right? And so in this case, we've got a gel that has a ladder in lane 1, and then it has 4 different samples, 4 different PCR reactions in it. And with those 3 to 4 things in mind, let's get going. So I'm going to close this first, and I'm going to open it in FIJI. So I already have FIJI open. When you open it on your computer, it's probably just going to look like a dialog that's up in the top. It's got a bunch of different tools here that you can use to do image analysis. We're only going to use one today, as you'll see. But the way you load your image is you can just drag it right in, and you'll see this drag and drop thing show up. And there we go. So now we've opened up our image in ImageJ. Okay, so let's go over to the spreadsheet here. There's a few different areas. One thing to keep in mind is you only need to modify the cells that are yellow in color. Okay, the rest are areas that will just auto-calculate. And let's get going here. So the general idea is that when you put DNA in a gel, there's a dye in there. The dye might be called red safe, or if you're old school, it might be ethidium bromide. And that dye will bind to DNA. And when it binds to the DNA, it fluoresces, right? And so the more fluorescing you have, or the brighter the band is, it typically means you have more DNA there. So the brightness of the band and the amount of DNA are tied together, okay? And so for today, we're going to use the ladder, which has a known amount of mass in it, to calibrate our spreadsheet. And then we're going to do some measurements around a band of interest to try to calculate the amount of DNA there, okay? And so a little bit more on this idea that there's a known amount of DNA within the ladder. So when you add ladder to a gel, as you hopefully have by now, you might add 5 microliters or 10 microliters of ladder. But generally, if that ladder was prepared properly, in that 5 or 10 microliters that you add to your wells, there will be about 500 nanograms of DNA there, okay? And you'll see here a bunch of different numbers for the mass of these different DNA fragments. And so, for example, you'll see here the 3 kilobase band. If you were to add that 500 nanograms of ladder to the gel, that 3 kilobase band should be about 125 nanograms of those 500. The 4 kilobase band here is only about 33 nanograms. The 1.5 kilobase band is about 36 nanograms, and so on and so forth. And so because we know what particular fragment in a gel has for amount of DNA, that's what we'll use to calibrate our spreadsheet here. Okay. And so I guess the only important thing here to note is you do need to add the right amount of DNA ladder to the gel in order to get a semi-accurate calculation at the end here. So for example, if you accidentally add 600 nanograms instead of 500 nanograms, these values are all going to be low, right? Because in 600 nanograms, you're going to have a lot more DNA there. So this is going to go higher. And if you don't input it correctly in the table, you're going to get weird numbers. So we'll assume when you ran your gel, you added about 500 nanograms to the ladder well, and that's what we're dealing with. So let me just pull this up again. And so it should look something like this under when you do the imaging. This should be about 500 nanograms of DNA here. Okay, so it should be clearly visible. Your band here, the 300 kilobase band, is brighter, as is in this image, so it does look fairly similar. And so we could assume that this band here, for example, has 125 nanograms. The 2 kilobase band underneath has got about 48 nanograms. And again, that's what we're going to use to calibrate. So how are we going to do that? Let's just start filling things in here. One thing to note, which is pretty important, is you should try not to use bands that look saturated, where the light looks saturated, because that means the pixels, they might be at the maximum brightness, and you lose information, because if it is actually supposed to be brighter than that, then it's not going to be accurate. And so I recommend that you use a bit of a dimmer band. This band is probably okay to use as well, but if you use a dimmer band, you know that it's brighter than the background, but also you haven't reached the brightness limit of that photo. All right, so all of these bands look pretty good. If your band is extremely bright, I strongly urge you not to use any filters if you're capturing these on your phone. The filters are going to adjust and falsify the pixel intensity. So try to use a raw image from your phone or whatever you're capturing it from. All right, so we're going to use this band here. I think that's a very nice band. Again, it's not saturated. So if we look here, let's start filling in the spreadsheet. So that's the 2000 base pair band. It has a mass of about 48 nanograms. This field here is asking how many microliters of your PCR reaction did you add to the gel? And you might recall you added 10 microliters to your gel, but when you made that initial cocktail, you only added probably 1, maybe 2 microliters, depending on the instructions. And so you probably added maybe 1 microliter of your PCR product, a couple of microliters of the 6x loading dye, and then more of just ultra water, right? So this is not asking how much did you add total to the gel. This is adding just your DNA, okay? And so it's probably around 1 microliter. All right, so those are filled in now. Let's go back to our loaded gel, and I'm actually going to reduce the size of this window a little bit, so make it a little easier. So now in this section, the idea here is to measure the brightness of this band, and that will help to calibrate how much brightness relates to how much DNA. And the way we're going to do that is first we want to collect some background signal. Now, what is background? So when we look at this gel, there is background light, right? Like there's brightness. The gel isn't black. If it was perfectly black, that would mean there's no brightness at all on that particular pixel within the image. But it's clear that there's brightness here. We've got a purple looking background. And when we look at this band here, we're seeing the DNA, but we also have to keep in mind that the background that we see here is also in this area. And so in order to just see the DNA and not this purple background mixed in, we need to kind of minus this background from the signal of the DNA. And the way we're going to do that is we're going to take little rectangles. And so you'll see I'm clicked on the rectangle here. And we're just going to collect some rectangle information surrounding, some brightness information surrounding the band of interest, okay? And so you draw a little rectangle. If you go to the top, you'll see Analyze, and then you can click Measure, okay? And the shortcut to do the measure is usually a Control or Command M. And that's what I'm going to use from now on after this time. So I'm going to click on Measure. And you're going to see a little dialog box pop up that says Results. And so this is the first measurement. It's telling me the area of this little rectangle is 248 pixels squared. The mean intensity or brightness, so this is a very important value for us right now. That's the kind of average brightness of the pixels across that whole area. And then it also gives us a minimum and a maximum, which we don't really need. Okay. And so, yeah, I'm going to go ahead, and if we look back over here on our spreadsheet, that's what we need to put. So this 113.903 goes into that there. I'm going to take another measurement. I'll just take one right underneath. And you want to be careful not to get any of that colored band when you're doing the backgrounds here. So I'm going to do that Command M, and you'll see another one pop up here where the mean is 104.226. There we go. And now let's actually do the band. So then this next row here is the actual band signal that we want. So I'm going to surround this band. I'm going to click off. I'm going to go a little bit bigger. You want to get all the signal. There we go. That's a little better. And you want to be pretty close to it. Something like that. Yeah, great. And so now I'm going to measure that. And we can see it pop up here, where the value is 128.122. And then you'll see one more field we need to add in for this band measurement, which is the area, okay? And the area here is 1632 pixels squared. Great. And so now we've got the spreadsheet calibrated, where it's done a little bit of calculating here to determine the total background, DNA minus background signal, and then it's calculated back using this value up here to get an idea of DNA signal per nanogram of DNA. But this doesn't really mean too much for us in general. You'll see. This will really depend on your image, the camera that was used, etc., the resolution. So yeah, this can be a little bit all over. So don't worry if your numbers are very different from someone else's. All right, so now we get to the bottom part here where we're actually measuring the DNA PCR product fragment, okay? And let's go ahead. For this demo, I'm gonna measure this band here. And I'm going to do the same thing, and we'll do this much quicker now. So I'm going to take two background measurements that are close by, background 1, background 2, and I'm just going to go straight into it and do the measurement of the band so that all the data is there and ready to go. Just need to make sure I input the right values because I have these past values as well. All right, so I've got 105.949. I've got 108.095, and then in the band measurement I've got 138.801, and the area of that is 1998. There we go. So now with all those measurements inputted, at the bottom here we can see the estimated mass of that DNA within the band. So it's suggesting here that the amount is 97.99 nanograms of DNA. And that makes sense. Generally, when you have a fairly bright band in a gel, it's about 100 nanograms. So that makes sense. It makes sense, okay? And so, yeah, that's the method here. You've used ImageJ to analyze an image of your gel, and you've got a pretty good approximation of how much DNA is there. It might be, 10-20% off, right? It's not super precise like a spectrophotometer, but you've got a pretty close estimation. And so I recommend you can go through and you could use these other bands. You could even do averaging of multiple measurements using different bands. And even check out the bright band. Again, bright bands, as they become saturated, the information and the calculation kind of gets thrown off. So maybe don't use this too often, but check it out, see what the output is, right? Great, so that concludes this video. Have fun measuring your gels, and hopefully you got some good amount of PCR product.